everyone, welcome to our second video about enzymes. This video is all about the characteristics of enzymes and how they work. So let's start with characteristics. I'm going to tell you about 5 different characteristics of enzymes. I'm going to show you a couple pictures and then ask you a couple questions about it. So first characteristic, enzymes do not make anything happen that couldn't happen on its own. It just makes it happen faster. So think back to that Batmobile from the earlier video. Batmobile could go to a race and cross the finish line on its own, but if it had an enzyme, which was shown by a Pac-Man in the Batmobile, it would happen faster. So enzymes don't make anything happen that couldn't happen on their own. They just make it faster. Second characteristic, enzymes are not used up in reactions. They can be reused over and over and over again. So basically, enzymes are recyclable. You can keep using them over and over and over until they become denatured, which we'll talk about in our third video. But typically think of enzymes being used over and over. Third characteristic, enzymes are only needed in small amounts. Since you can use them over and over and over again, you don't need a ton of them. It's kind of like the idea that if you bought a like Hydro Flask type of bottle, you can use that over and over and over again. You don't need to have that many Hydro Flask. Whereas if you just had, packages of plastic water bottles from Kroger, you would have to keep buying them over and over again because they won't last in the same way that a Hydro Flask would. All right. Characteristic number 4, each enzyme is highly selective about its substrate. I'm going to show you a picture and tell you something funny to help you remember that in just a minute. But know that each enzyme is highly selective. That means it's choosy about its substrate. Step 5, or characteristic 5, enzymes chemically recognize and bind to their substrates. So how do the enzymes know which substrates to connect with and help those substrates form their product? Well, it's a chemical thing. So certain molecules that are on the enzymes and the substrates help them bind together, and then those substrates can react with each other. All right, here comes some pictures that's just going to help you understand these concepts. Here is showing that enzymes, let's say sucrase, they have these active sites, and they can take a substrate that fits perfectly nicely into their active site and do a reaction. In this case, it's not actually 2 substrates coming together. It's one substrate that needs to get broken into 2 things. So sucrose comes into this active site, and the enzyme helps that sucrose molecule actually break into 2 separate molecules, which are fructose and glucose. Those 2 products, fructose and glucose, are released, and the enzyme can be used over and over again for as many sucroses come on in and need to be broken up into glucose and fructose. So this diagram shows how those enzymes are recyclable and they can be used in this cycle over and over and over again. Next up, we have this little picture, and this goes to show you that there are different enzymes that work with different substrates. So I kind of said that enzymes are specific to which substrates they'll work with, and they're kind of choosy about it. So you can see this enzyme, it has a certain chemical shape that would react with these substrates, not these other substrates, but these ones that are right there with it. They have the same shape in our pictures, though technically those shapes just represent the way that the molecules look, and those are microscopic. You wouldn't be able to see them. All right, enzyme type 2 here would work with a triangle-shaped substrate. It's choosy. It is not going to work with a circle-shaped substrate or a hexagon-shaped substrate. It's just going to work with the substrate that fits chemically with that enzyme. And type 3 works with this specific type of substrate. So here's the funny story that my students liked last year that helped them remember this. Let's imagine that I wanted to start dating somebody and I chose to do online dating. Not really my style, but let's just imagine. Let's imagine I'm a substrate, like this one over here, and I want to find a partner, like that one over there. Now, eventually, couldn't I meet somebody and, we would form a partnership, like a relationship on my own? Yeah, eventually I could. But if I wanted to speed that up, then I could use a dating site. And the enzyme here is kind of like the dating site. Now remember, I said enzymes are specific to their substrates. So there's a lot of different dating sites out there, right? I might have better luck with the enzyme or dating site Farmers Only than I would with Silver Singles, which is designed for an older population. So certain enzymes are designed for certain substrates, just like certain dating websites are designed for certain type of people. So kind of interesting, kind of funny little comparison there. Maybe it'll help you remember. All right, you ready for some questions? First, could the reaction that would take place still happen on its own without an enzyme? Yes, it could. It just would happen more slowly and it would take more energy. That would have a higher activation energy without an enzyme. So the reaction could still happen, but it would be slow and it would take a lot of energy. Next up, do enzymes get used up in reactions? No, they don't. They can be reused and recycled over and over and over and over and over again. Next, do you need a lot of enzymes to make reactions in the cell happen faster? No, because they are reused and recycled and they work pretty quickly. You do not need to have a lot of enzymes. You just need to have enough to get the reactions going. Next, can enzymes work with just any molecule? No. They can only work with specific substrates. Just like in my kind of funny analogy, different dating sites would only work for certain type of people. Different enzymes would only work with certain substrates. All right, here's how they work. First, they have the specific shape. Then part of the enzyme matches the shape of the molecule that's going to be reacted, and that molecule is called the substrate. Then part of the enzyme that binds to the substrate is called the active site. When the substrate and the enzyme bind temporarily to each other, the enzyme-substrate complex is formed, and you saw that in our first video. Then the activation energy needed for the reaction to occur is reduced. After the reaction is complete, the substrate has formed a new product, and the enzyme can be released and reused. Here's a little video to help you understand.
Enzymes enable molecules called substrates to undergo a chemical change to form new substances called products. Each enzyme acts on a specific molecule or set of molecules called substrates. Each substrate fits into an area of the enzyme called the active site. This fitting together is often compared to a lock and key mechanism.
Those are two
However,
substrates.
The enzyme changes shape a little to fit with the substrate. In the enzyme-substrate complex, the enzyme holds the substrate or substrates in a position where a reaction can occur easily. After reaction, the enzyme releases the products and can go on to carry out the same reaction again and again.